To justify this statement, I think I will take liberty and talk about the history of immunology, the major concepts, if we can call them, and see where this present work which fetched these three people, a lady and two gentlemen, one Japanese and two Americans, the coveted Nobel Prize. When we were doing PhD, immunology, we used to make fun that this is a witchcraft. That you didn't see immunology as part of a curriculum of a basic science department for a very, very, very long time. Madurai Kamaraj University had Muthu Karuppan, who worked on lizard immunology, and he introduced really proper cellular immunology in this country in the university system. Around 1986 or 87, I introduced immunology as a paper, specialization or elective course in Delhi University Zoology Department. Probably barring these two, no other university, no other department was teaching immunology as part of their postgraduate curriculum. Mostly it was in medical colleges. Somewhere starting with 1930s and 1940s, some chemistry people entered immunology. Michael Heidelberger, others. And therefore, some respectability came to immunology. And so people took it very seriously. And in fact, there were two groups of people, those who worked on the confusing cellular immunology and those who came from chemistry background, protein chemistry especially, and therefore they called it immunochemistry. And that's where the inroads came and made this field a little more quantitative, little more respectable. Otherwise, the terminology used in immunology is totally confusing. There is semantic jugglery. As we go along, we will notice, we'll explain all these things. Now, there are two terms here, and the very definition of this will make you laugh. One is called antigen, another is called antibody. And you ask, what is the definition? It is a circuitous definition. Antigen is one that binds antibody. Antibody is one that binds antigen. So I don't know where the wisdom comes. Difficult to understand this kind of a thing. However, as far as the record goes, the very first Nobel Prize, 1901, it went to von Behring. And what did he do? He took patients who recovered from diphtheria, took their blood, collected the serum, and then gave it to newly infected patients. And lo and behold, they recovered health, partially at least. And so serum became some sort of a magic drug those days. In fact, people were administering the so-called serum without knowing what is it doing, what does it contain. And it led to what we call today as serum sickness. Just liters of serum. All sorts of reactions happened. Later, when people like Elvin Kabat and others who used separation techniques and asked, what is this so-called antibody and where is it found and all that. In fact, there were two types of techniques that answered. One was an analytical ultracentrifuge. And people did this and said that in terms of Swedberg units, it was about 7s. So that is all the information that you have for the antibody. Similarly, they did the zone electrophoresis and got all the, if you take a drop of serum and do the electrophoresis, you will get in starting from the fastest pre-albumin, albumin, alpha, beta, gamma globulin. And they checked each band for this so-called antibody activity, some sort of a bioneutralization activity, and found that it fractionates or maps under gamma globulin fraction, the slowest moving. In fact, it doesn't move. It goes in the opposite side at pH 7, 8. So basically, therefore, some sort of a terminology came that antibodies are gamma globulins and gamma globulins are silent antibodies, another circuitous definition. And that's all we knew. Then came Landsteiner, who described blood groups. I don't go into detail for all this. And immunology suddenly became very respectable because blood transfusion was being given and even organ transplant and all that. You need to know this blood group, and so matching can be done. Around 1935, there was a guy called Strauss, and he asked a very simple question. If I take a well-characterized antigen, that means say a protein, in fact, he worked on egg albumin, and then mix it with this antibody, how did he get? You inject into a rabbit, and then after some time collect the blood and fractionate and get the serum. You call it antiserum. And so he took this antiserum, some amount, and then took that antigen, which was 10 egg ovalbumin. All you can do protein estimation and know exactly how much you are adding. So in a series of test tubes, he added graded quantities of ovalbumin, incubated with the fixed quantity of antiserum, and then after some time, he got a precipitate, takes 2 to 3 days, and then centrifuged and took the precipitate and estimated protein. Now, very simple experiment. This was done, and in 1959, my teacher, Maudgal, demonstrated that you can produce antibodies to hormones. It was unheard of at those days, because hormones are physiological things and mediators and essential for us. How can you have antibodies? It did. He demonstrated. And then in 1969, I took this antibodies to, at that time, luteinizing hormone, and did this quantitative precipitant test. Believe it or not, very simple data, protein estimation, because you know how much you added, we calculate molar ratios, we got the result that antibody is bivalent. Because if monoclonal antibody, you can, at those days, of course, there was no monoclonal, it doesn't give you cross-linking, therefore, simple molar ratios. And at that time, we also did a little bit experimentation and made a guess that you have only 4 to 5 epitopes in a protein like luteinizing hormone. No, that number was shocking. Now I will tell you something else, somebody else who did. There was a guy, immunologist, called Zuhair Abbasi. I will repeat now, he was a Muslim. There is a purpose for mentioning that. And what he did was, he cut the protein that he was handling into different peptides, immobilized them, and took the antiserum against the whole protein and poured onto this column where you have individual peptides immobilized. And then he found that the whole antibody can be exhausted by passing on to immobilized 5 or 6 peptides. That's all. Therefore, he came to a great conclusion. The number of determinants. Now, this determinant is the same as epitope. It all depends whether it is in the part of the protein or a separate. If it is a small molecular weight separate, you call it a hapten. So for the same thing, we have 3 names. Whatever it is, the whole thing, he was a Muslim. And why I'm mentioning this, at that time, there were big groups in Harvard who were, including some famous protein scientists, and who mentioned that these epitopes are continuous, and therefore there are infinite number of epitopes for a given antigen, a protein antigen. Of course, it essentially, believe me, those days, it was actually a fight between the Jews and the Muslims. They wrote dirty things in the papers that were published. Zohair Abbasi, Muslim, versus Israeli scientists who were working in so-called Tel Aviv, including Ephraim Kachalsky and others. So such serious dirty things happened in the field of immunology. They were fighting on a theory without taking into account their backgrounds. Anyway, leave it at that. So ultimately, we believe, in fact, the using computational biology and new software, 2019, that means 6 years back, it was proved that the number of epitopes are only limited 5 to 6. What we were talking for 50 years has been solved. This was one problem. The other problem was this. I also did work in reproductive biology. My boss was a reproductive biologist. And we know these mammals, when they have the baby, you've never seen a mother rejecting her fetus in pregnancy. There are exceptions, one or two. These are called threatened abortions. And this is one thing which puzzled people, because the fetus or the new growing fetus is a foreign. If you go by the classical paradigm of immunology, that you produce antibodies to foreign things, you don't produce it against self. Till today, nobody has defined what is this self. Is it in a physiological or philosophical way, or is it geographical or origin? No clarity on these things. Anyway, we all believe that you don't produce antibodies against your own antigens. When today we know self-antigen means it has arisen from your own body, one of the cells, either on the surface or soluble antigen, whatever it is. Foreign means another. But it can be same species, but still you call it foreign. So in that sense, therefore, this self and foreign became some sort of a major concept in immunology without understanding what exactly is foreign, what exactly is self. And then there were theories that there is a tissue or gland or organ called thymus. Below the sternum, you can find it. It's a major lymphoid organ. In addition, there is a bone marrow. Originally, this bone marrow, which was called as symbol as B, was used to refer to Bursa fabricius in birds. That we adopted. And so bone marrow cells are called B cells. Whatever it is, here comes the first theory. If you look at immunology, you don't see paradigms, concepts which are proven experimentally and which lead to new thinking and all that. Absolutely nothing. Everything is beliefs. So basically, here is an idea which people gave, that thymus being a lymphoid organ, and which is the one which is interacting with the so-called foreign antigen, there are cells which also react with self without defining what is that self, and that these are eliminated by apoptosis or programmed cell death. Now, there is an inherent contradiction in this sentence. That means these thymocytes, whose surface is usually having an immunoglobulin, and depending on the level of differentiation, it may be mostly IgM in the initial stages. And then later on, of course, others come. They recognized the cell antigen. If you call cell surface proteins in all our cells as self antigen, but they don't respond. Not only that, having stated they don't respond, they say that they undergo apoptosis. That is also a response. In other words, there was a semantic jugglery as to when they talk in immunology, reasons for certain phenomena. But all of us believed it, and everybody said yes, that those self-reacting thymocytes are eliminated, inducing apoptosis. Who does? The self-antigens. Now, this, in the present day, that is this year's Nobel laureates, they made a funny statement that low concentration of the self-antigen interacts with the thymocytes which have IgM surface and induce receptor downregulation followed by apoptosis, programmed cell death. Now, I still not able to understand this phrase low concentration. We have done, starting from my PhD and many others have done, lot of work on hormone receptors. In some cases, if you have high concentration of hormone, the receptor gets downregulated, gets internalized, just like in the case of thymocytes, which are seeing self-antigen. Moreover, self-antigen, as we understand, are the surface membrane protein. Mostly it is HLA and all. In mouse, you call it MHC. And therefore, it's a high concentration. It is not at all low. But these Nobel laureates, they have used specifically that low concentrations cause downregulation. And the other thing they have used, others have also used, and I have still not understood anything, that the state, this thymocytes who are supposed to react with self and who don't react, they are in a state of anergy. I looked in Oxford Dictionary, what is this anergy? Because in our work, we showed that in receptor downregulation and internalization, if you put respiration inhibitors, mitochondrial chain inhibitors, and inhibit oxygen uptake, you don't see that. Therefore, this saying that this thymus non-responsive cell is in an anergy state, I have never understood. But these this year's Nobel laureates have repeatedly used this term. Leave it. So now comes the question. Some of these thymocytes who have been eliminated by apoptosis, that is self-reacting cells, somehow escape that penalty and they are circulating. And then comes what we call, or these Nobel laureates are talking, peripheral immune tolerance. How does this happen? Again, contradiction in terms, semantic jugglery. They say that when the self-antigen meets this IgM containing thymocyte, or what we can call them as self-reacting lymphocytes or thymocytes particularly, receptor downregulation and what we call post-transcriptional RNA editing. Those of you who know the structure of immunoglobulin, you know there is a heavy chain, there is a light chain, that it's a polycistronic thing, and there is a RNA splicing and rearrangement and all that. You call it editing. Now, this thymocyte, which was self-reacting, as a response to this self-antigen, when they meet the local self means one of, if it is a mouse, it is MHA, MHC, or if it is a human, it's a HLA, class 2, class 1, all this. That they undergo not just apoptosis for the central mechanism, in the peripheral immune response, they are circulating and they undergo this what we call as they call it receptor editing, which means RNA splicing occurs post-transcriptional event. And instead of producing an antibody which reacts with the self, during rearrangement, they produce an antibody which reacts with foreign. Now, these terms are very easy to know. How do they know the foreign when they have not yet met? This is what the Nobel laureates are saying. Okay, that's one confusion. That means one, low concentration causing this down regulation, but the data for all other systems show it is high concentration. Everybody has agreed on this thing. And the second thing is this receptor editing, where RNA splicing occurs and new type of receptor or immunoglobulin is produced, which does not react with cell, but reacts with foreign. How does this happen? I have not understood, because how does the thymocyte know who is foreign, who is self? All right. So I leave it at that. But then it has other ramifications. This not interacting with self-antigen is not a rule. For example, many people suffer from autoimmune disorders. For example, diabetes type 1, you see circulating antibodies to beta cells of islets of Langerhans. Similarly, Hashimoto's thyroiditis. Like that, there are many. Lupus erythematosus is a very famous one, where DNA antibodies are found, your own DNA. And therefore, this self-foreign is not very clear in immunological language, because you do see autoimmune disorders, many people, and if you get one autoimmune disorder, you are bound to get within your lifespan many other types of autoimmune disorders. That means it is genetic basis. Now, in that sense, therefore, how do you get these antibodies? If you say that you don't react with self-antigen in this so-called central immune tolerance, how do you get these autoimmune disorders at all? There is no clear explanation in the literature, including the Nobel laureates' work. Now, this valency problem has people thought they have solved it. This happened in mid 1970s. All of you know that Caesar Milstein demonstrated first time monoclonal antibodies, but then actually you get a monoclonal if you take a polyclonal producing cells and dilute it to one cell. But he clubbed it with another one, which is called somatic cell hybridization, where he combined these mouse myeloma cells, which are perennial, that means eternal or they don't die, cancer cells, and clubbed them or hybridized them with the antibody producing spleen cells. Now here, in those days, he used Sendai virus. Today we use polyethylene glycol for this cell-cell fusion. So in that sense, when he produced monoclonal antibodies, people realized that it does not form a precipitate like normal antibodies who are polyclonal antibodies. The reason is very simple. Cross-linking is not possible, and therefore precipitate is not formed. However, immediately what happened was these monoclonal antibodies found applications, tremendous applications, and that's how most of immunology work, if you see most famous work, it never clarified any immunological concept, but a lot of applications were found, both antibodies as drugs, monoclonals for therapy, anti-cancer therapy, and other things. In other words, immunology, unlike other areas of biology, has no independent set of concepts which have been experimentally verifiable and which denote the domain area, the boundaries of this subject. There is no such thing here. Whereas immunochemistry, dealing essentially as a protein chemistry, in fact, I think in CFTRI, MS Narsingrao used to operate a analytical ultracentrifuge. Probably there were only 2 machines in the whole country in those days. Anyway, so the whole point I'm saying is that immunochemistry became more respectable because everything was quantitative and you can talk chemistry, you can talk physics, you can do everything. Whereas cellular immunology was still very confusing, even identifying. For example, these people, current Nobel laureates, have discovered something called regulatory T cells. I don't know how many of you practice immunology or read and teach immunology. In our days, there were 2 types of T cells, the T helper cells, which present the antigen after processing, and the T suppressor cells. Nowadays, nobody talks about T suppressor cells. I have never understood this mystery. The regulatory T cells that these Nobel laureates are talking, are they T suppressor cells? We don't know. And they have not come up clear with any clarity in all these things. So therefore, but no doubt it's a great contribution they have made in analyzing the mechanism of this peripheral immune tolerance. Our gut microbiome, for example, is totally foreign, but we don't produce antibodies to our gut microbes. Powerful antigenic proteins are there. We don't produce, and we don't know the mechanism of this. This is another thing that we tolerate. So both the extremes, that you can produce antibodies to self-antigens, that you need not produce antibodies to foreign antigens, both can be proved, both are true. And the contribution of this current this year's Nobel laureates is in explaining peripheral immune tolerance. Of course, they have ascribed reasons which are contestable, but nevertheless, it sounds okay. So either through centrally eliminating by apoptosis the so-called self-reacting thymocytes, or in the case of peripheral thing, you do receptor editing and produce from IgM another type of immunoglobulin which does not react with the cell. This is what the papers are saying as the major contribution of these 3 people, especially the major contribution came from Sakaguchi, the Japanese fellow. One experiment which they have reported and which we don't understand again, if you remove thymus cells before a certain critical age, you will notice lot of self-reacting T lymphocytes in circulation. It almost creates havoc. Whereas after some age, if you remove this, nothing will happen. That means thymic education, where that you should not react with cell, has occurred at a critical window during embryonic development or during ontogen. And that itself is a major contribution conceptually from this Nobel laureates. Now, I can go on like this, but I would better field questions because, as you notice, cellular immunology has no rigor. Only immunochemistry has a rigor. But let us see whether some of the questions that you are going to ask me brings clarity to myself. Let us see.
So the
Can I
floor
start?
is open. Yeah, floor is open. Any one of you can start raising points.
Don't make it a quiz program.
So can I ask?
Yeah, please go
Please,
ahead.
sir,
Yeah, it's
for my clarity.
more like something to add. I find that the one part is the recognition by the receptor and the ligand, whether we call it an antigen, antibody, whatever.
Absolutely.
The other part is actually the stimulation of the cellular proliferation. A particular
Yeah,
type of cell is allowed to proliferate by a signaling pathway which is mediated through the growth
absolutely.
factor
Absolutely.
like pathway, and
The
that
title of this,
actually sort of
yeah,
amplifies the effect.
correct, absolutely correct. The title of this abstract that I gave was molecular recognition. No details are found. In fact, they have found a protein which is found in this regulatory T cells, and they have not said to what it binds, to what it recognizes. How does it suppress self-reacting thymocytes? Where does it bind? No clarity. I'm not belittling their Nobel Prize, but there is no clarity in all this work. You are right. It has to bind, it has to recognize first, and then all the intracellular events can take place. Most important, proliferation.
But I couldn't understand that these people say if the T regulatory cells can be, levels can be raised, you can have a cure for, the autoimmune disease. What is your comment on that?
Okay, number, very frankly, I have not done any work in this area.
Yeah, but
The whole point is, see, when you say you can increase or decrease some cell, whatever type of cell, you have to recognize what are the properties of that cell. They have not said anything other than saying there is a peculiar protein. I forget the name of it.
FOXP3. FOXP3.
FOXP3.
Mm.
By just incorporating FOXP3, something does not become a regulatory T cell. We have to know the total cellular antigen profile and cell typing, as we call. That I don't think they have done. For example, cluster differentiation markers, we know for many immunocompetent cells, but they have not said anything on these so-called regulatory T cells. CD markers.
In fact, when Macfarlane Budner talked about self and non-self, how is his ideas different from what these people are talking?
I don't know whether he talked as self and non-self. What he formulated was clonal selection theory.
Correct.
Fantastic.
Which was always said as self and non-self.
Yeah, it's a very fantastic, because opposed to him were giants like Linus Pauling,
Yes.
who said that no, the antigen instructs the antibody to fold.
Fold, correct.
There is nothing called selection. So for about 40 years, people were fighting selection theories versus instruction theories. Leave it. So in the clonal selection theory, the antigen which has entered the body picks up the particular cell which has got an antibody in a low concentration to shoot himself or shoot the antigen itself. In other words, you select the gun with which you can commit suicide. That is the meaning of clonal selection theory. And people didn't understand that how do you choose your own death warrant? Others don't react, only that react. So these are the strange phrases that you see in immunology, cellular immunology.
Okay. But coming to this, in fact, I have the same issue when we talk about protein degradation. And the ubiquitin goes and binds to the protein to be degraded.
Yes, yes.
But how does ubiquitin know that I have to go and sit over there? I mean, nobody, same issue like antigen antibody, what instructs them to recognize? So I find what you are raising is, this is a question I also used to ask myself, how does ubiquitin actually recognize? Then they say there is E1 enzyme, E2, they made a complex story. But the fact is, how does it recognize?
Some other signal from that cell must be coming, which ubiquitin recognizes. That we don't know the detail. Therefore, we could talk only in circuitous logic like this.
Yes.
Same thing for example, anamnestic response, secondary immune response. How do you get why some antigens can induce but some other antigens don't? These explanations have not been given, actually. Cellular immunology is very, very vague. I'm not belittling the contribution of these 3 Nobel laureates. All I'm saying is that it leaves more
questions than
unanswered
answers.
questions but it opens new areas. Therefore, people.
I think most important probably in this work is that Japanese fellow correlating with the genetic studies and arriving at FOXP3. The story
Absolutely.
developed further. I
No,
think
that's a major discovery, I would say, but
yeah,
it does not clarify anything.
correct.
Because where does this FOXP3 bind to what? How does it interact with the thymocyte? All these things are not clear at all. Of course, I don't say they have to finish the story in one go,
Yeah.
but it certainly raised more questions than answered anything.
But Professor Murli, I would like to ask you if an youngster wants to choose a problem on these kind of areas, because everybody is playing for immediate success. But the kind of problems you are raising is unsolved. And
Yeah.
ideally, as a youngster, should look at unsolved problem, not which is already in the process of, lead is already there. So what is your advice? What are the questions a child should ask? Like you said, how does it know it is foreign? It is this thing. How does it know?
These are all terms. Of course, there are two answers for you.
Mm.
You recollect in your own field, Perutz.
Yes.
So when he joined Bragg, he said solve the structure of hemoglobin. Now,
Correct.
where is there a technique for that? Absolutely. You give an open-ended problem for PhD,
Yes.
right?
Correct.
So, like this, basically, you pick up a problem. All that we can say is ask a question which has not been asked by others.
Mm.
However simple it is, that is what we call original. If you can ask original questions, if it is too tough for you to answer, fine, leave it. But if you can answer that by simple, elegant experiments, then I think that is more worthy of doing original work. And that is what our PhD students should be told to do. However, see, the PhD production is, there are different traditions in different institutions. In a typical British type of thing, you leave the student to find himself everything, come up with a question and then answer it. But in most of the cases in Indian universities, it is a guided work. Practically, you hold the hand of the student and tell the experiments you have to do, when to finish. And if the great student is willing to work, you will finish. Now, therefore, there is no scope for doing original work.
Mm.
Because you need a system for that, a culture. Then only you tackle. That's how
Correct.
Perutz took 19 years for that, but then he solved so many problems. No?
Correct.
That's what I'm saying. Let us be very realistic. PhD is a training program. If you can, how to ask a question, how to know what is not known in that area of research, and then skills. For example, experimental skills have to be taught. People forget it. In fact, if you see the syllabus of most of the MSc biology related thing, you see how many people conduct experiments in immunology. Is hardly is there. Science is not poetry. No? Therefore, the more skills you know in terms of experimental experience, you can work on PhD, good PhD, great PhD, and all that. But our culture doesn't permit all that. We have a lot of internal problems. That's why we don't produce original great work. At best, somebody makes a observation and leaves it for somebody else to do the work.
Correct. Anybody else would like to ask questions? Feel free.
Sir, I would like to add one thing that I feel as a student of, biology in immunology. I find three concepts are interesting. One is the allelic exclusion, another is VDJ recombination,
Correct.
the third one is clonal selection.
Absolutely. All are very vague, but then they're like an orchestra, they happen.
Yeah.
It's
At least
allelic exclusion as well as recombination.
yeah,
Who is directing all these things?
like the
How are the
VDJ,
sites
yeah,
identified?
at
There
least the
doesn't seem to be clarity.
yeah, at least the VDJ recombination could be created by transfecting the RAG1, RAG2.
Yeah, no. I know more details about it. All I'm saying is,
that is just
it looks like an
controlling
orchestra.
factor.
One after another, it happens, and you get an assembled immunoglobulin. But it's really, when you talk in terms of, let's say, philosophical level, who is directing in all these things? How do you expect something and then do accordingly? Metabolism is adjusted for that. This we call it ontology in science. That you want to do that, therefore it happens. This kind of logic, but it doesn't work. There's no purpose in all that happens in our physiology. But we talk as though because you have to fight the enemy, you have to produce an antibody. This kind of, we are caught up in semantic confusion completely. That is why biology is very, very tough to understand. And immunology, of all the areas of biology, is the worst of that.
Any other questions or any doubts? Feel free to shoot. It is a informal session only.
Yeah. Surinder.
Sir, I just want to make a general comment, if you allow me.
Yes, sure.
Who am I not to allow?
See, here what students are asking, once we do PhD, do we get a job? So they are not asking the basic fundamental questions. That is, forget about the basic fundamental questions. Where does this PhD leads me to? So this is another question now. It has bothering most of the students. So from that point of view, how to reorient this whole discussion on PhD?
There are two things I will do. In one of the Indian Social Science Congress, which is a parallel to our great Science Congress, I was asked to deliver some B.V. Rangarao memorial lecture. And I have analyzed the sociology of doing science. It all depends on traditions. Why some institutions do good work, why others don't do good work. More important, if you do the sociological analysis, 90% of people who have come to science or to higher education in general are from middle class. And therefore, the first thing that I look for is, do I get a job with that? You are not talking about answering some great question and doing earth-shaking research. There is no such thing. It is business. What do I get if I do PhD? Will I get a lecturer job? Because of this mentality, I'm not blaming anybody. I'm also from middle class. There was not even an appetite for wanting to do some good work, some original work and all. There was no such thing. Of course, the guides were terrible also, some guides. And therefore, you need a culture of original work and inculcate that culture. One way to do that is, of course, it can boomerang. You give financial support to that student till you finish PhD. Don't worry, I'll give you. Of course, some people have taken it as an employment. University of Hyderabad, I know many examples. So there is a danger in doing all that, because you can't do great research unless there is this tension, this pressure that I have to submit. I have to finish so much work. If that is all eliminated, at least guide and the student can think of some new work and then get PhD will come. Whatever you do, PhD will come. I remember in my PhD days, there was a colleague, I won't tell the name. What is a PhD? 49 figures and 32 tables. That's all. You fill it up, you get PhD. There was a famous guide who used to tell that. What I'm saying is, we have become silly, actually. Nobody is discussing seriously, profoundly thinking, analyzing what are we doing. And you have from the government also, the feelers that you get. Oh, China is producing 30,000 PhDs, we are not producing. I don't know why you should ape what China is doing or Thailand is doing or America is doing. Do you have a worthwhile problem to pursue? Are you competent to analyze it? You have, of course, infrastructure, because modern biology work, you need infrastructure. You can't do research on Mall Road in Delhi. Therefore, all that is understand. But given all that with monetary support and all, still people took to, at best, there is a sociologist called Vishwanathan, Shiv Vishwanathan. Some of you might be knowing. All that we do at best is replicating Western science. So it is like, confirm what others have done. But the culture, unless everybody is disagreeing with that, there's no way to do original work. Everybody is doing. I'll take 2 more minutes just to seriousness of this problem. About 2-3 months back, I was in Orissa. They have some Biju Patnaik award or some such thing. And I was chairman of the committee to decide all those for all branches. Each committee convener was proposing some names. So the engineering committee fellow came up with a name. I am from biology. And then in numbers he says is 1 lakh citations, 165 H index. And it went on like that. I said, stop, I'll give you the award, don't worry. But tell me, can you explain what this work is to a layman? He couldn't. He was always crunching the numbers, that's all. What exactly is the work? Nothing. Einstein also remarked it. If you can't talk science to your mother, explain, is not worth that science. That means clarity of thinking has to be there. What is important, what is not important, what is original, what is new, what is not new. These things have to be very clear in our minds. Then everybody will do good work. Sorry, I don't know whether I answered your things or not. In fact, I had a slide which shows somebody who did PhD went up, Yamaraj, and that Chitragupta said, you have done a lot of bad things, so I need to punish you. But then I hear you did a PhD, that is good enough, he said. It's hell.
Muthamani, you want to ask a question, Muthamani?
No, actually, I'm most privileged to join this group for the first time. I really appreciate Professor's view on what is research in the present days. It's only a number. Wherever we go, it speaks about the numbers. So actually, I got a project from DRDO recently, and I was searching for JRF, and I advertised in many forums like social media and all those, and I received only 6 applications. So the problem is, I asked them to join PhD with me, but they say, no, we cannot join PhD. We can work as a JRF or project assistants. That means that they're not interested. They're interested only on money. So I don't know where we are going with this kind of status. Like when we, take statistics about 10 years ago and 20 years ago, the dedication of students for pursuing PhD is just going down. So if that is the case, and in India, we are producing and we are aiming at producing a number of PhDs, numbers. We want more numbers, and how much is the quality, and what, as Professor said, what we contributed to science is yet a big question. So that's what you people know as seniors, and we look forward wherever you go and wherever you participate, and inculcate that kind of culture to bloom again. So that is what my opinion is here.
Yeah. We have a question from Malaysia. NT, did you please kindly go ahead? NT Johnny. Yeah.
Thank you, Prof. Kanan, Prof. Muralidhar. Sorry for if I'm mistakenly pronounced. I would like to ask, and maybe that this is kind of silly question maybe. Actually, I'm not an immunologist. I'm not really familiar with all the pathways of those cells in immunology, but now I'm working on MDR, multi-drug resistance organism, which is called Acinetobacter baumannii, and see how is the interaction between this bacteria with the host innate immunity with the patient's blood serum and plasma from the patients. So my question is whether you have any experience on the things that you said before at the beginning regarding the anti-serum or the serum sickness phenomena. As I know, this kind of serum sickness usually happens to most of them is a gram-positive bacteria like Clostridium or Corynebacterium diphtheriae. But as we know, as a human, actually we already equipped by God a set of cells, either it's T cells or B cells and or other cells, including in the immunology in our human body, normal human body. But I think the most part of the the rule, I mean the variable is the antigen itself. Maybe you have any experience kind of serum sickness applied, but other than gram positive, perhaps any other species or genus, why cannot we apply this model to another type of antigen, for example, like virus or any other type of bacteria, gram negative or even the mycoplasma or other things that, especially for the multi-drug resistance, maybe you have any experience on it. Thank you, Prof.
Honestly, I don't have an experience in microbial related immunology. I've never worked on microbes. My work is mostly on hormone antibodies and all that. So the answer to your question is I don't have experience in that area, so I cannot comment anything on that.
Okay, thank you, Prof. Okay, thank you.
Yeah, but we'll circulate this. Anybody else would like to answer her question? Anybody else? Surinder, you wanted to ask something? Surendra.
Yes. No, I'm just learning. I'm just learning. Nothing.
Okay.
All of us are learning.
That's a good. There's a question in the question box. No, one question is there. If we abstract immunology down to core or basic principles, is the immune system fundamentally an antigen recognition system or a context-dependent decision-making system that uses antigens, danger signals, and tissue state as inputs to decide between tolerance, inflammation, and repair? This is a question asked by Balakrishnan.
Yeah, you can interpret it in another way. Go to old physiology, there is something called homeostasis. So we have a system of cells producing antibody and other competent cells and all that. The entry of an antigen causes disturbance. And therefore, the system tries to fight back to get back to its old equilibrium, steady state, not equilibrium. And therefore, that is my understanding of this whole immune response and all that. Sometimes it is alarming, and so you have bystander effect, lot of damage is done. Sometimes it is good because it also gives you protection. But we have, if you permit, I will show those 3 slides.
Yeah, I can
where
share.
If you do chemistry, you will convert that problem into a very elegant problem and lot of insights you can get.
I'll just share that one for you. Yeah.
Just the 2 slides is enough.
Yeah.
I'm sure this is for self-aggrandizement. I will tell what we did.
Yeah.
Anyway, thank you so much for this very interesting discussion. It really gives me like such enlightened me And
I should
know,
thank you for coming and listening.
Yeah.
I know this
Thank
even
you.
from my supervisor, Prof. Suba, maybe you know her, Prof. Suba Basu. Thank you.
But if you register in our site, whenever talks happen, you can suggest what you want. I could arrange talks on those lines. Okay.
Okay, thank you.
So kindly register for me to send you notification. Can you see, Murli?
Yeah, I can see.
Okay. Yeah, now you can go ahead.
Can I operate from here? No, I can.
No, I'll do it. You just tell me.
Okay. As I had remarked earlier, instead of basic immunology has given a lot of byproducts in terms of technologies and all, because this is part of health research. And we also did a little bit antibodies as drugs. Next slide.
Yeah.
See, what is depicted here is a funny thing. It's a line diagram of a molecule, a hormone called human chorionic gonadotropin, which has two subunits, and this is representing one subunit of that, beta. Why I am putting this is, if you notice there, this beta subunit has got about 6 disulfide bonds. We controlled the rate of reaction by dithioerythritol and knocked out one by one. That means 1 disulfide, 2 disulfide, 3, 4, 5, 6. At that point, when we arrived at DS5 HCG beta, that means 5 disulfides are knocked out, only leaving one of them between a cysteine 9 and cysteine 90. What is so great about this? This became an immunogen which produced antibodies highly specific to HCG. So what is great in that? I'll take 2-3 minutes. We had a great economist called VKRV Rao, long back, Nehru days. He was later education minister also. He brainwashed Nehru and proposed that population size is a drag on economic growth. And Nehru believed it. From that day, India was the first country to formulate what we call policy on family planning. And therefore, they diverted a lot of funds for people to do work on contraceptives. Because we knew a little bit of antibodies, we developed new immunocontraceptives. One of them was this DS5 HCG beta, elegant chemically reduced and alkylated. Otherwise, when you reduce this disulfide bond like Jarasandha, the protein will fold back. Air can oxidize. And therefore, we tied up that sulfidyl group with iodoacetylide, so it cannot fold back. But we got that one epitope, and I was just mentioning, next slide, that the antibodies do not cross-react with human LH, which is a pituitary origin, which is similar to HCG. Why I am mentioning this? See, there are two things which all of us know. Libido for man and the disturbance of menstrual cycle for woman, they cause a lot of mental stress. And therefore, any immunocontraceptive that people develop should not affect menstrual cycle. In the initial days, people left, right, center, in fact, Australians, Americans, Vernon Stevens and others, they used this molecule and produced antibodies and recommended it as a vaccine, contraceptive vaccine. But the data shows a terrible cross-reaction, and therefore those women have psychological problem. What we did was simple chemistry and showed that this DS5 HCG beta has no cross-reaction with HLH and therefore should not disturb menstrual cycle and cause psychological stress to the lady. Of course, we have not done human trials, but we showed it very clearly, as this is a radioimmunosec curve. And the top right-hand portion shows all other hormones don't react, only HCG reacts. So in other words, what I'm saying is, immunology is full of semantic confusion. But if you apply physics and chemistry and do some elegant experiment, you can still get products which are applicable to humans. There's no problem on that. This is one example. The best so-called immunocontraceptive vaccine candidate we produced in our work. Of course, I have not done any human trials, so I don't know that.
In fact, Professor Talwar tried to do quite a bit on this, no?
I don't want to open my mouth on that.
Okay.
It's not good, actually.
No, I agree. I agree. I mean,
Lot of unethical things.
Yes, yes, I agree.
See, in phase 1 clinical trials, he went to Punjab, took 6 postmenopausal women, gave them HCG shots, whole HCG, and told everybody, look, they're not pregnant. Postmenopausal women. Why do you need a vaccine? Anyway, it became a laughing stock. We do a lot of unethical work. I think I don't want to talk on these things.
I agree. Totally. In fact, Professor Muralidhar has written a book on ethics also. You know, with a lot of people.
Sense of publication.
Yeah.
Because Ajkal, as one in the audience also mentioned about, I think, Muttumani. See, there is a lot of craze for awards, recognitions, and all that. I said disease is because we are middle class. Middle class, even if you commit murder, somebody has to say sabash.
Yes.
Then we are happy.
Yes.
So in that, there are a lot of unethical scientific work being done. Highly irreproducible, plagiarism. ICMR has sent out that nowadays the young scientists in ICMR institutions, 28 of them are there, they're repeating what ICMR did in 1920 or 1930, the initial days, and then calling it as a new project. That level of,
unethical practices.
Unethical and dishonesty has crept into these premier institutions. One of my students is a deputy director somewhere, and he was telling that this is what young people are doing. Just take old annual reports and repeat it. I think we have most unethical country is India. We are not fit for science, actually. There is no intellectual honesty.
I think we have spent more than an hour. Murli, I'm sure the audience must be quite tired. If there are no more questions, I would like to, okay, somebody is asking some questions. Yeah, Sridhar, please speak. Sridhar, yeah.
Sir, good evening, sir. As usual, it's very interesting. See, at last point, that the intellectual honesty, what needs to be done? What is the way forward?
Nothing can be done. See, preaching doesn't help. This comes from inside, actually. So, actually, your journey starts with your PhD. So, what kind of science culture, ethical culture, all that picks up from that first lab. And subsequently, of course, depending on stature. So if that influence is a corrupting influence, then the whole life is gone. That's what happens.
The problem of getting an experiment right or wrong, how to get rid of that fear of reporting a wrong result to a supervisor?
Because you will waste your time in reproducing those irreproducible results. There are many, that there is a journal and as well as people who watch that and report, but it's a tiring, why should you do that? At the cost of, take a risk. I'll tell something about, which don't quote me, don't publicize all this. There was a vice chancellor of Mysore University called Rangappa. Tremendous plagiarism, unethical thing, lot of things. Chemistry. He had DST projects on this, all false. Finally, somebody complained and DSTs asked me, please inquire and give us a report. So I went to DST, sat there 2 days, read his papers and all that. Unadulterated plagiarism. Lifted papers from Osmania University Chemistry Department, changed the authors, published it. When we were doing all this and I was writing the report, I got the DST person who is assisting me, he got a call from the biggest scientist in this country, don't touch Rangaswamy. I resigned. I told, never call me for all this kind of a thing. And the next year, this person becomes president of Indian Science Congress. That is India. So I don't like, this is all useless. It's not good also. It's very unfortunate. So a person who works in such labs, you can see what he will imbibe. See?
Exactly.
Please don't quote me. This is actual thing. You can go to DST and see the records, all those things.
No, another very sad thing these days is number of years a student takes to finish his or her PhD. 10 years, 12 years, without fellowship, the kind of harassment which happens is very unfortunate.
So-called guides have no clue what they're doing, what they're guiding. Totally incompetent people call themselves guides, which is not correct. See, at worst, PhD is a training program.
Yeah.
At best, yes, luck gives and everything else works out, you will turn out a great work.
Yeah.
There's nothing wrong in doing great work, but don't harass the student. He's not a labor. He's an intellectual partner, younger in age. Therefore, you will have to mentor them.
Correct.
In my article, which is, some of you, I'll send the article, which is published also. The number of suicides that you see on PhD students is pathetic.
Yes.
Why the nation we are not waking up on these things? That's why I went to that sociological congress, science congress, and gave this lecture and all that. With data, I showed them that. Very, very unfortunate. I'm telling you, younger people are running away from science because there's nothing too exciting them. They think this is all bogus.
I agree. Yeah. I think we will put if there are no more questions, I would like to thank Professor Muralidhar for this, very informal, but taking us through what immunology is and, what are his perspective. I would like to thank all of you for taking time off to be with us. And
I should thank Kanna for giving me the platform and all of you who came here. I don't know whether you got enriched or not, but I love talking like this because sometimes we have to be very informal to reveal the insights. All these things. So don't quote. I don't want any court case or anything. It doesn't matter. Yeah.
No, but I want also that, you feel free to contact me on my website. Anybody interested in a particular topic to be organized where you would like to have an expert, I'll try my best to get somebody from anywhere in the world because I'm fortunate to have many people all over the world. So it'll be my pleasure. And please continue joining us so that, we would like to continue this.
But Kanna, I think you remember very well. Yeah. You remember the BEST workshops we used to conduct?
Yes. Yeah.
I think you should revive that. Take all the topics in biology in a broad sense, conceptual topic, arrange for good speaker. I think it will become a very good supplementary education.
In fact, what Professor Muralidhar is talking, when we were in Hyderabad, so myself, Murli, Dipankar, Sushil Dubey, all of us got together and we said we had all wanted to help the PhD student. Those days, PhD was much tougher because students didn't have an access to using a spectrophotometer. I have attended seminar, in those conferences we learned, and we used to have it 3 days before the SBCI meeting, Society of Biological Chemists. Then the president got so annoyed that everybody comes for the best program and goes off, doesn't attend the meeting. So he said, no more SBCI meeting. This kind of program we don't want. It happened. So we had to close it in 1989. We started it in 83 and 89, it found it natural. But I think it's good suggestion. If others are interested, we could revive that.
We brought out volumes also, 7 volumes.
Yes, we brought all the
written articles are there.
Yes. Monica, you want to ask something? Monica? Yeah, Monica, please go ahead.
I'm trying to unmute myself. Yeah.
Yeah, sure.
Excellent discussion, sir. I feel privileged to be a part of this this discussion today. I'm happy and I'm feeling blessed that I've got an opportunity to meet Professor Muralidhar Garu. So I hope he remembers me. I was with him in proteomics facility at University of Hyderabad, SLS. So all his guidance I tried to take lots and lots of his suggestions and advices. So it's just a thank note that I wanted to give him. I'm always grateful to you, sir, for your.
No grateful. Just
Thank
to
you very
lighten the mood, I'll tell you a joke. Now that you mentioned.
So
In
thank you.
one of the centenary or platinum jubilee of Indian Institute of Science somewhere, they called Sidney Brenner to give a talk. And then they asked Obaid Siddiqui to introduce the speaker. Typical Siddiqui went on and on and on. At the end, he said, request Sidney Brenner to come onto the stage and give the lecture. Brenner said one sentence. The lecture is over, you can ask questions. Then we had a DG of CSIR, brags a lot. So he said, we have done this, that omics, this omics and all. Sidney Brenner asked him and shut his mouth. The best omics is economics. We should be humble when we are doing work.
Correct.
That's the problem.
Anyway, thank you so much, Professor Muralidhar, for, thanks to
Thanks,
you,
sir.
so many people joined. And I thank all the people who joined. And please keep in touch. You can register in kkbiotech.net. And whatever help you want, we'll be more than happy to provide. Because a lot of people want to help people. But
Absolutely.
people have to come to us to ask if we want this help. So thank you all once again and all the best. Thank you so much, Muralidhar.
Thank you, Kanna. Thank you, everybody, for your patience.
Yeah.
Thank you,
Thank you,
sir,
sir.
Professor Muralidhar, Professor Kannan. Thank you so much.
Yeah.
Array, nahi. Why do you thank? We are all
No, sir.
professional teachers.
Yeah.
So it was very, very interesting and very encouraging to also